The overall goal of this procedure is to visualize and quantitate chlamydial inclusion growth dynamics in living cells. This is accomplished by first generating fluorescent host cells that stably express a fluorescent protein, such as GFP, in their cytoplasm. Next, the host cells are infected with chlamydia and allowed to incubate. Then, the infected cells are visualized by live fluorescence microscopy. Finally, chlamydial inclusion characteristics are quantified through microscope analysis software. Ultimately, live fluorescence microscopy is used to visualize chlamydial inclusions within the host cell and to measure biological parameters of inclusions for quantitative results.
The main advantages of this technique over existing methods, like antibody-based enumeration, are that this reverse imaging strategy can be used on live cells and also in collaboration with other fluorescent labels to quantitate inclusion dynamics of all chlamydial species. Furthermore, unlike routine methods that label bacteria and cells, this approach illuminates the inclusion membrane in living cells and can thus reveal important dynamic properties of this pathogen-containing compartment.
After generating fluorescent HeLa cells according to the text protocol, plate GFP HeLa cells on a desired culture vessel, such as glass-bottom dishes for high-resolution imaging, or 24-well plates for IFU determination and multi-well screening. Thaw a frozen tube containing C. trachomatis or any other desired chlamydial strain on ice and dilute in HBSS to the desired multiplicity of infection. To perform a static infection with C. trachomatis LGV serovar L2, aspirate the media from the wells and use HBSS to wash the GFP HeLa cells.